Hello, everyone. This is Dr. Richard Fleming, and today is Monday, the 21st of December, 2020. We now have two vaccines approved by the FDA for emergency use authorization, with more questions today than we had a month ago. As I've emphasized in some of the recent videos, the EUA documents provided to the FDA and the publications do not provide measured information about T cell responses or antibody responses to the vaccines. These are critical pieces of information to understand the true safety and efficacy of either the Pfizer or Moderna vaccine or any other vaccine that comes along. Social media shows people with money trying to get the vaccines first, and senior physicians being vaccinated before junior physicians, with junior physicians complaining. It would appear that some of our least appealing qualities as humans are surfacing in this pandemic. Perhaps one should not be quite so eager to be in the front of the line, given the lack of evidence and published evidence about antibodies and T cell responses. The purpose of being a scientist is to look at things objectively and ask scientific questions and then answer them, or at least attempt to. If you've read any of the material I put on Amazon.com, particularly part 1 of Unmasking COVID, where I go through the process of how viruses infect people and then get cells in our bodies to make more viruses, you'll recognize that when viruses enter the cell, there's several different genetic components. One of those is called, for SARS-CoV-2 or COVID-19, is called the open reading frame 1. That genetic information provides the blueprints and subsequent enzymes to get our cells to make more of the virus. If the only mRNA included in these vaccines are the structural proteins for the S protein spike itself, which we now know produces antibodies to the N-terminal domain of that spike protein, as I've talked about in prior videos. And we now know that those antibodies increase the ability of the S spike protein to infect cells. In addition to the knowledge that the vaccines themselves will increase the ability of the virus to infect cells, producing an inflammatory thrombotic response that's been associated with morbidity, mortality, harm, and death to patients, we need to ask the question, how do our cells, once injected with these vaccines, know to make the virus? If the only part of the mRNA, the messenger ribonucleic acid of the virus that's being injected into people is for the S spike protein, how do our cells know to make the S spike protein? The instructions for that are in another part of the genetic material of the virus in the open reading frame 1. So, if in fact we have a self-amplifying mRNA, that's the term for it, then that genetic information must also be part of what's being injected into people. What alarms me is why the committees who were responsible for looking at the safety and efficacy of these vaccines did not ask these fundamental questions. They're important questions, and they're about truth, transparency, benefit, and risk, boiling down to the bottom line question of, are these vaccines safe and effective? Sometimes it seems that the information being provided to us is being pushed so rapidly as to get by the necessary questions that need to be answered. As a scientist, these are the questions that need to be answered, because these S spike proteins can't reproduce themselves within our cells unless we're also adding other parts of the virus into the vaccines to tell our cells to make SARS-CoV-2. Just more questions for people to consider, along with the fact that in a couple of videos past, I speculated on the possibility that since we don't know what the mRNA is that's included in the vaccines, and since we know that SARS-CoV-2 is the result of at least two human modifications, raising the question about whether the vaccine is in fact SARS-CoV-3, one of the people that have been listening to the channel and other social media sites raised the confirming comments that there's a new strain of SARS-CoV-2 or COVID-19 going around the UK. Interestingly enough, this strain of the virus only appeared after the vaccine program in the UK began, raising questions once again, is the vaccine being promoted really SARS-CoV-3?